We have already studied different checkpoints of the cell cycle. One was at the end of G1 phase, other was at the end of the G2, and the third one was at the middle of the M phase. But there are several other genes are also involved in the checkpoints. For example, if a DNA gets damaged, then the cell need to be all at that point of the cell cycle. You know why? If the cell progresses into the next phase of the cell cycle, then after the cell cycle, there may be several progenies or several gametes may be formed. All those gametes or all those progenies of that parent cell might have bear the same mutations in the DNA. That means those kinds of the mutations may get amplified. So these are detrimental. So need to prevent the inheritance of that mutation into the progeny cells at any cost. So these kinds of DNA damages are tackled in different ways. For example, if the DNA damage that occurs in the G1 phase of the cell cycle, then that damage recruits the proteins like MRC11, RAD50, NB51. So these proteins gets assembles over here and forms mRNA complex. M means MRC11, R means RAD50, and N means NB51. So this protein gets assembles over here and forms mRNA complex. That mRNA complex recruits another protein that may be known as ATM dimer. This ATM monomers having kinases activity. So each of these monomers that phosphorylates another monomers. That means a kind of autophosphorylation. This autophosphorylation causes the monomerization of that dimers. Then each monomers of that ATM gets binds with the NB51. So the monomer of the NTM that binds with the NB51 that already assembled over here, that may be part of the mRNA complex now. So this binding causes the phosphorylation of H2A histones. This phosphorylation of the histone protein that causes the disassembly of these histones from that area. Normally we all know that the histone that causes the curling of the DNA so that the DNA gets condensed over here. So here what may be happened is that histones get phosphorylated by these ATM proteins. That phosphorylation causes the disassembly of that histones from this area. This area means from the DNA damaged area. So this causes the other proteins like CHK2 that can access the DNA damaged area. After the removal of the histones from this area, the secondary structure of this DNA over here gets disassociates, and now the DNA damages can be revealed. Now the proteins like CHK2 that can be joined over here, and then the CHK2 that gets phosphorylated, and that causes the phosphorylation of the other protein that is known as P50. Then that P53 causes the phosphorylation of another protein that is P21. The P21 that inhibits the CDK. CDK means that inhibits the CDK2. We all know that the CDK2 along with the cyclin E and cyclin A that drives the cell cycle from G1 to S phase, not G to S phase. So what may be happen here is that ATM phosphorylates P53, P53 phosphorylates or activates the P21 proteins, P21 proteins that blocks or inhibits this CDK2. So what happened here is that the cell cycle drive from G1 to S phase that may be blocked over here because the CDK2 are now inhibited. So These cyclins E and A cannot be get activated. As a resultant of that, G1 to S phase of the cell cycle that will be get blocked. Not G2, this is S phase. So once again, the DNA damages during G1 phase of the cell cycle that recruits the proteins like MRC11, RAD50, NB51 over here in the DNA damaged area. So they form a complex is known as mRNA complex. Then that mRNA complex that recruits another protein, that is ATM dimer. Monomers of this ATM are or having kinesis activity, so they gets phosphorylated each other. That means a kind of autophosphorylation. Then each of the monomers of the ATM that binds with the NB51 of the mRNA complex, then the ATM that causes the phosphorylation of another protein, that is H2A. H2A means it's a component of the histone complex. Then the histone complex gets disassociated from here. So the DNA damaged area that gets revealed. So another proteins like CHK2 that can access over here. So it can bind over here, the CHK2. Then that CHK2 that activates another protein, that is P53. That P indicates protein, 53 indicates the size of the protein. This is 53 kilodalton or 53,000. That is the molecular weight of that protein. So P means this is a protein, and 53 means that's molecular weight of the protein. So this CHK2 that activates the P53, so it gets phosphorylated. The phosphorylated form of the P53 that cannot interact with the proteasomes. Normally, the non-phosphorylated form of the P53 that can interact with the proteasomes. But here, after the phosphorylation, it cannot interact with the proteasomes. So this P53 cannot be get degraded. Then the level of the P53 gets increased in the cell. Then that phosphorylates another protein, that is P21. That P indicates protein, and this number that indicates the molecular weight of that protein. Here it is 21, so the molecular weight of the protein is 21,000 or 21 kilodalton. So here the CHK2 that phosphorylates P53. So phosphorylated form of the P53 that cannot interact with the proteasomes. Then the level of P53 gets increased. Then that P53 that activates another protein, that is P21. So it gets phosphorylated. Then that phosphorylated form of the P21 that inhibits the CDK. Here we are now discussing about the G1 phase of the cell cycle. So here the CDK is the CDK2 that we have already studied, that CDK2 along with cyclin A and cyclin E that drives the cell cycle from G1 to S phase of the cell cycle. Here the CDK2 that gets inhibited, so the cell cycle drive from the G1 to S phase that gets blocked or arrested. So the cell cycle that's stuck at the G1 phase of the cell cycle. So by that time, different DNA repair enzymes that can recognize as these DNA damaged area, and damages gets repaired. Then these assemblies gets disassociated, and finally the cell cycle that maybe progresses from G1 to S phase of the cell cycle. So the importance of this regulation is that by stucking or stopping the cell cycle at the G1 during the DNA damages, then the repair enzymes will get ample time to repair these kinds of DNA damages. Otherwise, the cell cycle that may progress into next phase, that is S phase, there what may be happen is that DNA gets duplicated, and then cell may be drive into G2, then M phase, then finally then that cell is getting divided into its own progenies. Damages in that single cell that get amplified into different progeny cells.